In continuation of the last video of paper Chemistry of Life of MSc Semester 3, in this video we will study the structure and function of coenzyme A. Coenzyme A has complex structure consisting of an adenosine triphosphate, a pantothenic acid which is a B vitamin, and cysteamine. The coenzyme is involved in the transfer of acyl groups. The sulfhydryl group, that is SH, of the cysteamine moiety of this coenzyme forms a thioester with the carboxyl group of the acyl compound, such as acetic acid, to produce the acetyl coenzyme A, which is one of the most important coenzyme A derivative. The thioester bond is energy rich and can easily transfer the acetyl group to an acceptor. Coenzyme A or CoA or SHCoA or CoASH is a coenzyme notable for its role in the synthesis and oxidation of fatty acids and the oxidation of pyruvate in the citric acid cycle. All genomes sequenced to date encode enzymes that use coenzyme A as a substrate, and around 4% of cellular enzymes use it or a thioester as a substrate. In humans, coenzyme A biosynthesis requires cysteine pantothenate, vitamin B5, and adenosine triphosphate ATP. In its acetyl form, coenzyme A is a highly versatile molecule serving metabolic functions in both the anabolic and catabolic pathways. Acetyl coenzyme A is utilized in the post-translational regulation and allosteric regulation of pyruvate dehydrogenase and carboxylase to maintain and support the partition of pyruvate synthesis and degradation. In 1953, Fritz Lipton won the Nobel Prize in Physiology or Medicine for his discovery of coenzyme A and its importance for intermediary metabolism. If we consider the functions, the first is fatty acid synthesis. Since coenzyme A is in chemical terms a thiol, it can react with carboxylic acids to form thioesters, thus functioning as an acyl group carrier. It assists in transferring fatty acids from the cytoplasm to mitochondria. A molecule of coenzyme A carrying an acyl group is also referred to as acyl coenzyme A. When coenzyme A is not attached to acyl group, it is usually referred to as CoASH or HSCoA. This process facilitates the production of fatty acids in cell's membrane structure. Coenzyme A is also the source of the phosphopentothene group that is added as prosthetic group to proteins such as acyl carrier protein and formyl tetrahydrofolate dehydrogenase. So if we see the plan, then we see that vitamin B5 is the precursor of coenzyme A and it functions in three ways. In the one way, it forms as a cell group carrier and takes part in the fatty acid synthesis and oxidation. In the second manner, it forms acetyl-CoA derivative and takes part in citric acid cycle and produces energy. In the third manner, it behaves as a regulator in lipid metabolism. Then we come to the energy production by coenzyme A. Coenzyme A is one of the 5 crucial coenzymes that are necessary in the reaction mechanism of the citric acid cycle. Its acetyl coenzyme A form is the primary input in the citric acid cycle and is obtained from glycolysis, amino acid metabolism, and fatty acid beta oxidation. This process is the body's primary catabolic pathway and is essential in breaking down the building blocks of the cell such as carbohydrates, amino acids, and lipids. Now we come to its regulation activity. When there is excess glucose, coenzyme A is used in the cytosol, and cytosol is known as intracellular fluid or cytoplasmic matrix or ground plasma, and this cytosol is the liquid found inside cells. The cytosol is a crowded solution of many different types of molecules that occupy up to 30% of the cytoplasmic volume. So here we see that coenzyme A is used in the cytosol for synthesis of fatty acids. This process is implemented by regulation of acetyl coenzyme A carboxylase, which catalyzes the committed step in fatty acids synthesis. Insulin stimulates acetyl coenzyme A carboxylase while epinephrine and glucose inhibits its activity. During cell starvation, coenzyme A is synthesized and transports fatty acids in the cytosol to the mitochondria. Here, acetyl coenzyme A is generated for the oxidation and energy production. In the citric acid cycle, coenzyme A works as an allosteric regulator in the stimulation of the enzyme pyruvate dehydrogenase. If we consider allosteric regulation is the regulation of an enzyme by binding an effector molecule at a site other than the enzyme's active site. And if we consider what are effector molecules, then in biochemistry, an effector molecule is usually a small molecule that selectively binds to proteins and regulates its biological activity. In this manner, the effector molecules act as ligands and that can increase or decrease the enzyme activity, that is gene expression or cell signaling, thus acting as activator or inhibitor. For example, allosteric effectors, cytoplasmic effectors. And the meaning of cell signaling is the process of communication occurring between the cells and within the cells. New research has found that protein coagulation plays an important role in regulation of the oxidative stress response. Protein coagulation plays a similar role to S-glutathione in the cell and prevents the irreversible oxidation of the thiol group in cysteine on the surface of cellular proteins, while also regulating enzymatic activity in response to oxidative or metabolic stress. Now we come to the structure and function of vitamin B12. Vitamin B12, also known as cobalamin, is a water-soluble vitamin involved in the metabolism of every cell of the human body. It is one of the 8 B vitamins. It is a cofactor in DNA synthesis and in both fatty acid and amino acid metabolism. It is particularly important in the normal functioning nervous system via its role in the synthesis of myelin and in the maturation of developing red blood cells in the bone marrow. Myelin is a lipid-rich or fatty substance that surrounds the nerve cells' axons. Axons are nervous system's wires to insulate them and increase the rate at which electrical impulses called action potential are passed along the axon. If we come to the structure, then vitamin B12 is the largest and the most structurally complex vitamin. The vitamin exists in 4 near identical chemical forms, that is vitamins: cyanocobalamin, hydroxocobalamin, adenosylcobalamin, and methylcobalamin. Cyanocobalamin and hydroxocobalamin are used to prevent or treat vitamin deficiency. Once absorbed, they are converted into adenosylcobalamin and methylcobalamin, which are the forms that perform physiological activity. All forms of vitamin B12 contain the biochemically rare element cobalt, positioned in the center of a coronal ring. Coronal ring resembles the porphyrin ring which occurs in hemoglobin. Both feature form pyrrole-like subunits organized into a ring with a largely conjugated structure alternating double bond and single bond. The only organism to produce vitamin B12 are certain bacteria and archaea. Bacteria are formed on plants that herbivores eat. They are taken into the animal's digestive system, proliferate and form part of their permanent gut flora, producing vitamin B12 internally. Now we come to what are the sources of vitamin B12. Animal source foods: meat, fish, fowl, milk products and eggs. Grain-based foods have to be fortified by having the vitamin added to them. Vitamin B supplements are available as single or multivitamin tablets or intramuscular injection, or certain mushrooms also contain vitamin B12. If we come to the functions, then vitamin B12 is a nutrient that helps keep the body's nerve and blood cells healthy and helps make DNA, the genetic material in all cells. Vitamin B12 also helps prevent a type of anemia that makes people tired and weak. Two steps are required for the body to absorb vitamin B12 from food. First, hydrochloric acid in the stomach separates vitamin B12 from the protein to which vitamin B12 is attached in the food, and in the second step, vitamin B12 combines with the protein made by the stomach called intrinsic factor and is absorbed by the body. Some people have pernicious anemia, a condition in which they cannot intrinsic factor. As a result, they have trouble absorbing vitamin B12 from all foods and dietary supplements. Vitamin B12 deficiency causes tiredness, weakness, constipation, loss of appetite, weight loss, and megaloblastic anemia. Nerve problems such as numbness and tingling in the hands and feet can also occur. Other symptoms of vitamin B12 deficiency include problems with balance, depression, confusion, dementia, poor memory, and soreness of the mouth and tongue. Vitamin B12 deficiency can damage the nervous system even in people who don't have anemia. Vitamin B12 works closely with vitamin B9, that is folate or folic acid, to help make red blood cells and to help iron work better in body. Folate and vitamin B12 work together to produce as adenosylmethionine SAMe, a compound that is involved in immune functions and mood elevation. Vitamin B12, B6 and B9 work together to control blood levels of the amino acid homocysteine. High levels of homocysteine are associated with heart disease. Vitamin B12 is crucial in the normal functions of the brain and the nervous system. The metabolism of every cell in the body depends on vitamin B12 as it plays an important part in the synthesis of fatty acids and energy production. Vitamin B12 enables the release of energy by helping the human body absorb folic acid. That's all for today. Thank you.